What's going on, guys? Today we are going to start a new concept that honestly is going to be fairly familiar due to everything we've already talked about so far this semester. We've spent a lot of time looking at DNA, what's DNA's function, what DNA looks like, and how DNA really controls different processes. Today we're going to dig a little deeper into that and look at how DNA makes proteins. So proteins is obviously one of our macromolecules. It's one of the macromolecules that we haven't spent a ton of time on this semester or this year because this unit really is where we find it the most. Proteins obviously are very, very important. They make things happen in your cells. A good way to think about proteins when we talked about them originally, we had said that they're good for structure and function of different things. They truly are the workers of the cell. They do everything. So your DNA is the code. Your proteins are the things that actually make things happen. And that's pretty much the case all throughout your cells. Proteins are doing basically every function that there is to do in a cell. So to start off, we obviously need to make sure we understand DNA really, really well. I think we're all in a point where DNA is pretty good with us, but I do want everyone to just pause right here. And I want you to watch this video real quick. It's just going to kind of be a refresher, kind of set up everything that we're going to talk about next. So go ahead and pause this, watch this video, and then come back to me. All right, welcome back. Again, DNA is very important. We need to understand DNA pretty well to really get this next unit of RNA and protein synthesis. So just to kind of recap a little bit, DNA is deoxyribonucleic acid. That is the namesake of DNA, right? DNA, okay, deoxyribonucleic acid. It is a nucleic acid, which is one of our macromolecules. And DNA is really what makes everything in your cell function. It's the code that makes everything in your cell. Now, the one thing that we probably haven't mentioned about DNA that is going to be really important for this unit is this idea right here, that DNA can never leave the nucleus. Okay, we obviously know that it is in the nucleus, but it can never leave. It's stuck there. It cannot escape it. So when we talked about DNA, I'm going to throw up another picture here. That's not what I want. Here we go. So when we've looked at DNA, this is just kind of a good refresher. Okay, DNA obviously is a molecule that exists in a double helix form. The reason it's a double helix is so that it can really, really tightly compact and fit within the nucleus. So as this really tightly compacts, it forms almost like a rod shape, and that is obviously what we see as our chromosomes when we look at images of DNA and stuff like that. When we look really close into this molecule of DNA, we know that a molecule consists of two strands. We have one strand on the left, one strand on the right, and these strands are made up of nucleotides. Nucleotides are phosphates, sugars, and bases. When we've talked about DNA, there's only four bases that we've mentioned, and that is A, T, C, and G. As we move into this next part, that's going to be a little bit different. But for DNA, it will always be A, T, C, and G. Just a little closer look at our nucleotide. Okay, this is going to be, again, a structure we need to know for RNA, which we're going to introduce right now. It's going to be the same exact structure. The only thing that's going to be different is one of these guys, is the nitrogen bases that exist within this nucleotide. Okay, so let me jump back over here. So when we think about DNA, okay, obviously we start with a strand, and then we have a complementary strand. The complementary strand is the other strand that bonds together with that original strand to make the molecule. So if I'm looking at a DNA strand that is A, T, T, C, 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 G, A, C, T, C, C, if I were to ask you to write the DNA complementary strand, you guys know how to do that. You're simply just going to write it as the opposite base that would be on the other strand of that molecule. So the complementary strand A would be T, complementary to T would be A, so it would be TAA, GGG, CTG, AGG. Make sense? Now into this guy RNA. Okay, so DNA is a nucleic acid, as is RNA. RNA is ribonucleic acid. So what's the difference? Well, instead of deoxyribose being the sugar in DNA, in RNA the sugar is ribose. So the name of it designates the sugar that makes it up. Okay, so when we look at that structure of a nucleotide, the sugar in the middle is not going to be deoxyribose, it's going to be ribose. Now, RNA has one purpose, and one purpose only. Its purpose is to make proteins. It is the protein maker. Okay, that is its function in our cells. That's what it does. That's its full-time job. Okay, and unlike DNA, RNA can leave the nucleus, and that's really important when we look at how these RNA structures make proteins. Okay, so DNA is stuck in the nucleus. RNA can escape. It can leave. So again, looking at our nucleotide structure, okay, it's very, very, very similar with RNA as it is in DNA. We have a phosphate group that's attached to a sugar. Okay, this sugar is ribose, not deoxyribose. And then we have a nitrogen base. Now, the nitrogenous bases are mostly the same except for one. We have A, which is adenine, G, which is guanine, C, which is cytosine, and then there is no thymine whatsoever. Instead, we have this guy called uracil, which is U. Okay, so from now on, when we talk about RNA, we need to make sure that we recognize that U will always bond with A. G will still always bond with C. So I'm going to ask you to pause this really quickly, and I want you to copy this table down in your notes. This is a very easy way to remember everything that we just talked through. The one additional thing that we can mention here that I didn't previously is this first part, in that DNA we obviously know is double-stranded. There are two strands that make up a molecule, whereas in RNA there's only one strand. It's a single strand of nucleotides, and you'll see why that's important a little bit later. So pause this, go ahead and copy this down. This will be a good section of your notes to have. All right, so I just want to look at this idea of base pairing one more time. So DNA, obviously we already did that. So looking at this ATTCCGACTCC, that would be the original strand. So then if I asked you what would be the DNA complementary strand, you would say TAA, GGG, CT, GA, GG. Okay, but then something else that I can ask you is, what is the RNA complementary strand? Okay, so what you're gonna end up seeing happen is RNA is built off of a DNA strand. So we have to see what the DNA is first and then build the complementary RNA strand from that. Okay, and we'll get way more into that a little bit later. But if we look at the complementary strand, the rules work the same, except there's no thymine whatsoever. So A would bond with U, uracil. T would pair with A, T, A, G, 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 C, U, G, A, G, G. So notice that whenever you see a T, it's still going to code for an A on the complementary strand, but when you see the A, it's going to code for the U. So that we can skip. I'm going to ask you to pause this here, okay? I'm going to ask you to write this in your notebooks, okay? And then go ahead and write the complementary strand of RNA from this sequence of nucleotides. Okay, hopefully you got that correct. So again, we're going to look at this. We have G, so that's C, G to C, A to U, T to A. Okay, so double check what you have written in your notebook. Okay, hopefully you wrote the right complementary strand. If you didn't, go ahead and correct it. Okay, it will take some time to get used to that mix-up of letters with the DNA to RNA. All right, so I'm going to ask you to write this in your notebooks now. Okay, we're just going to practice one more time. So write this in your notebooks, pause this, write this down, and then this next slide I'm going to throw up a couple questions for you to work through in your notebooks. Okay, so here's that same sequence here. So A, C, T, T, C, A, G, C, T, A, blah, 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 so on and so forth. Okay, what I want you to do is underneath that, I want you to label DNA, complementary DNA strand, and then write the complementary DNA strand. Then write complementary RNA strand, write the complementary RNA strand. Okay, this last part now we're going to get into right now, but obviously it's not going to make full sense right now because we haven't really talked about the process, but I at least want to introduce it here. Okay, so go ahead and pause this, finish 1 and 2, and then I'll come back and we'll do number 3. Okay, very good. So hopefully you have that correct. Now I want to talk about this. Okay, so the purpose of RNA is to make proteins. Okay, so the DNA gives the code to the RNA, and then the RNA is what codes for the proteins to be made. Okay, now in this, when we have an RNA strand, so I'm going to go back a couple slides here. So when we have an RNA strand, it's segmented into 3 nucleotides at a time. Okay, these are what we call codons. Codons. Okay, 3 letters is a codon, and those 3 letters code for a specific amino acid. Okay, now if you think back, amino acids are the the monomers of proteins. Okay, so amino acids make up the proteins when you put a bunch of them together. So if you think about this, each of these codons codes for an amino acid, and when those amino acids are bonded together, they make a long protein. And then those are the things that actually function within our cells. Okay, so you will be given, and they're actually up on the front table, a table that looks something like this. That looks like this. Okay, so on this, you're looking at a bunch of letters, obviously. Okay, but this is how we determine what protein a codon codes for. Okay, I'm going to hopefully not confuse you through this. Okay, but I'm going to kind of jump back and forth between these PowerPoints here. Let me jump back here. So let's just look at this one, for instance. Okay, so CCU is the first codon in this RNA strand. CCU. So here's how you use this table. You go to this table, and we start in the center. Okay, we find the first letter of the codon. So we have GUAC. C was our first letter in the codon. It was CCU, remember. So we're going to go C. And then if you notice, this is broken up into quadrants, right? So we have this bottom corner, this top right corner, top left corner, bottom left corner. Well, the C encompasses this whole bottom right corner. So the first letter is C. So then I'm going to go to the second letter, which was C. So see these 4 letters here that I can choose from in this quadrant. So C, C. And then if you continue where C is at, you see the 4 bases again. And the last letter was U. So C, C, U. That codon makes the amino acid proline. So then the first amino acid in that protein is going to be proline. So then if we look at the second codon, which is, sorry, let me get back here, AAU, we find A here in the center,